Okay, so the next several problems deal with blood typing. So I'll work one example and then you should be able to work the rest of them from there. So with blood typing, you have your A, AB, B, and O, so those are our phenotypes, but our genotypes are represented differently. The alleles, you can have a capital I with an A, that superscript denotes the allelic form. That codes for type A. Then you have capital I with a B, that codes for type B. And then a little I codes for type O. Keep in mind that A and B are codominant, so if you have one of both, they'll both show up. So this problem asks you to show the possible genotypes and phenotypes of the offspring from a cross between a homozygous male with type A and a homozygous female with type B. So I like to kind of figure out what information I'm working with and then make my list of genotypes and phenotypes. Okay, so if we are looking at our homozygous male with type A blood, let's put him over here. Okay, so homozygous means that the alleles are going to be the same. So since he's type A, we're going to have capital I with A, capital I with A. Okay, so that's our male. Then we have our female. She is homozygous with type B. Again, homozygous, you're going to have two of the same allelic forms again. So since she's type B, you're going to have capital I with a B and capital I with a B. So now we're going to make a cross between these two. So we'll set up our Punnett square. Remember, you've got to make gametes, so you have to simulate meiosis. So we'll put our female on top. So we have one allele here, one allele there. So we have our B alleles. Then we have our male. And since they're both homozygous dominant for their traits, they just have the same allelic forms, and then we'll cross them. So you get I with B, capital I with A, capital I with a B, for all of them for this particular cross. So if we're looking at our offspring, you can list them however. I tend to make a chart form. So the only genotype we got was capital IA, capital IB, and this codes for type AB blood. So you can write it 4 times because that's what you got 4 times, but essentially this is all you're going to get every single time. Okay, so here's another blood typing problem. So this one says a man with blood type A, one of whose parents had blood type O, marries a woman with blood type AB. What are the genotypes of the offspring? So before you can make a cross, you need to know what your parents are. So if we look at our male, we know he's blood type A, but we know his parents, one of his parents had blood type O. So remember, you get one allele from one parent and one from the other. So we know he has at least one allele, one lowercase allele to show the recessive O. Because if you remember from our previous problem, capital I, capital A codes for A, capital I, capital B codes for type B, and then a little I, lowercase I, codes for O because it's recessive to both A and B. Okay, so remember he's going to have 2 alleles, so we know one of them is a little I, and to make him blood type A, the other one's going to have to be capital I, capital A. So that takes care of our man with blood type A. So then let's look at our woman with blood type AB. Okay, so we know she's AB. The only way to get AB, remember those are codominant, so you're going to have capital IA and capital IB. So one allele for A, one allele for B. They're codominant, and when they're together, they make blood type AB. So now we are ready to cross them. So if we set up our Punnett square, hopefully I drew that big enough. If not, bear with me. Okay, so we'll put our female on top. So one allele for each column. We'll put our male on the side. Capital I, little I. And we'll make the cross green just because. Okay, So you have capital I, capital A, capital I, capital A, capital IA, IB, capital IA, lowercase I, capital IB, lowercase I. Okay, and this one just asks you the genotypes of the offspring. So if you want to put phenotypes with it, great. I think it's a good practice, but you don't necessarily have to. So we have capital IA, capital IA. We have capital A, capital IA, capital IB. We have capital IA, lowercase I, and capital IB, lowercase I. If it asked you for the phenotypes, you could tell me that. So since you have two alleles for A, that one's type A. Since you have an A and a B, this one's going to be type AB. You have an A and a little I, so it's still type A because A is dominant to O. That capital I is dominant to the lowercase I. And then you have capital IB and lowercase I. Again, this allele is dominant to our recessive allele, so then you end up with type B. Okay, so this is one of the most commonly asked about genetics problems. So 41 says, in a certain species of plants, flower color is determined by a single gene. There's 3 different alleles. You have white, which is a capital P with a W, orange, capital P with a O, and red, which is a little p. Remember that P is going to denote your gene, and the superscript denotes the allelic form. Remember, capital letters are dominant, lowercase is recessive. It says the white and orange alleles exhibit incomplete dominance with the heterozygote, making pale orange. So if you have an orange and a white allele, it's going to make pale orange. The red allele is recessive to both the white and orange alleles. A plant with orange flowers is crossed with a plant with white flowers, and the phenotypic ratio is 1 to 1 to 1 to 1. So 1 pale orange, 1 orange, 1 white, 1 red. So this one you're going to fill out backwards to figure out the parental genotypes. So I recommend working with what you know. So I like to write out whatever my alleles are going to be, and then that helps me denote stuff. So big P with a W is white. We know that big P with an O is going to be orange. And we know that little p is going to be red. Let me find my red marker. That's okay, we'll just use this pink. And our problem also tells us that we can get pale orange if we have a big P with a O and a big P with a W. So if you kind of work from the inside out, we'll be able to get our parental genotypes. So to make a red flower, the only way we can get red is if we have 2 recessive alleles, because red is recessive to white and orange. So the only way we're going to get red, we're going to have 2 of the lowercase p's. So we automatically know we can kind of look in our columns and our rows. So we know red's going to have 2 lowercase p's. White and orange, we could have maybe a big P with a W and a little p. That could make white. We could also have big P with an O and a little p. That could make an orange. So you can have homozygous dominant or heterozygous dominant. So we don't really know what our white and orange are yet. But we do know pale orange because it said that it shows incomplete dominance if you have an orange allele and a white allele. So we know that we're going to have a big P with an O and a big P with a W. Okay, so you know that your columns are going to share one allele and your rows are going to share one allele. So we know for a white flower, we have to have at least 1 capital P with a W. Okay, so if our row is going to share an allele, the only other option is a lowercase p, because we can still have a white phenotype even if it's heterozygous dominant. Okay, and then so if we look at our orange, so we know our row is going to share one and our column's going to share one. So to have at least, we know at least that one of those is going to be a capital P with an O, because you have to have that to at least be anywhere near orange. And so they're sharing that P with the O together. And then our orange and our red are going to share an allele, because remember, our columns are going to share alleles. So the only possibility there is, again, heterozygous dominance, because capital P with the O and little p still makes the phenotypic orange. Okay, so then you can fill in your gametes around based on what the columns and rows share. So if we start at the top, our pale orange and our white, they both share a capital P with a W. And our next column, our orange and red, both share our lowercase p. So then our genotype of our white parent is going to be capital P with a W, lowercase p. So we're going to do the same thing with our orange flower. If you look in the top row, they both share that capital P with an O. Our bottom row, they both share that lowercase p. And the phenotype for the parent, or the genotype for the parent, is capital P-O, little p. So this last problem here is another one I get a lot of questions about. So this is a sex-linked problem. So a red-green color blindness is inherited as an X-linked recessive. So you have to have two of it for that to phenotypically show up. So if you only have one allele, if you only have one of these alleles, you're just a carrier, you're not actually colorblind. Okay, so if a colorblind man marries a woman who is heterozygous for normal vision, what would be the expected phenotypes of their children with reference to this character? So to get the phenotypes, we're going to have to get the genotypes of their children. But before we can get the genotypes of their children, we need to get the genotypes of our parents. So let's take our colorblind man. So we have our colorblind man. He's colorblind. Okay, so we know that if you're a man, you're going to have an X and a Y. But he's colorblind, so this X is going to have the little C superscript to denote that the colorblind allele is present. And again, you have to have, if you have one X only, if you have one copy of that and it has the colorblind allele, you are going to be colorblind. Then our female is heterozygous for normal vision. Okay, so she is normal, but she's heterozygous. So females, we're going to have two X's, but only one of those is going to carry that X-linked recessive allele. So now that we have our parents, we are ready to make a cross. Get your Punnett square. Okay, so we'll put our male on the side, put our female on top, and we'll do the inside in blue. Okay, so when you cross these, you get an XXC, XC, XC, XY, XCY. Okay. So let's list our genotypes and phenotypes. Okay, so the first one we have is XXC. We only have one allele present in this female, so we know it's, you can say girl, normal, or you could say carrier. Either one is fine in this case because you can't look at somebody and tell they're a carrier. So phenotypically she's normal. Okay, our second one, we have XCXC. Since it's a girl and there's two copies of this allele present, she is going to be colorblind. Our next one is a boy. He does not carry that allele, so he has normal vision. And then our last one is another boy, and he carries this allele, so he is going to be colorblind. So hopefully this helps you, give you some guidance with some of these genetics problems. If you have any more questions, I'll be online with Kaltura during office hours and lab time. If you're watching this on YouTube, feel free to like and subscribe. Again, email me if you have any questions.